Two parents, both with brown eyes. And then their child arrives. With blue eyes. Where did that come from? Neither parent shows it, and yet there it is, undeniable, written in the child's face. This is not magic. This is not an accident. It is genetics, and a simple grid called a Punnett square predicts it exactly. By the end of this video, you will know how to set up a Punnett square, read it, calculate probabilities from it, and avoid the 3 mistakes that cost students points every single year. Let's start at the beginning. In the 1860s, an Austrian monk named Gregor Mendel grew pea plants in his monastery garden in Brno. He was meticulous, obsessive even. He tracked 7 different traits across thousands of plants and dozens of generations. And what he found changed biology forever. Mendel discovered that traits were not blended together in offspring, the way most people assumed. They were inherited as discrete, separate units in predictable mathematical ratios. He published his findings in 1866. The scientific world ignored him for 35 years. When researchers finally rediscovered Mendel's work around 1900, they realized he had cracked the code of inheritance. He had died in 1884. He never knew his name would become the foundation of modern genetics. A few years after Mendel's rediscovery, a British geneticist named Reginald Punnett created the tool we still use today. He was trying to explain Mendel's ratios to skeptical colleagues quickly and visually. So he drew a simple grid. It worked. We still call it the Punnett square. Before you can build one, you need 8 vocabulary words. These words will appear on every genetics question you ever face. Let us go through them now, one at a time, clearly. Do not skip this section. The mistakes almost always happen here. Word 1. Gene. A gene is a specific segment of DNA that codes for a particular characteristic. Eye color, blood type, height, flower color. Think of a gene as the instruction manual for one specific feature of a living thing. Word 2. Allele. Most genes come in more than one version. Each version is called an allele. The gene says, determine eye color. The allele says, which color? Brown or blue. You inherit one allele from your mother and one from your father, so you always carry 2 alleles for every gene. Word 3. Dominant. When 2 different alleles are present in the same individual, the dominant allele is the one that actually shows in the physical appearance. We always write dominant alleles as uppercase letters. Capital B for brown eyes, if brown is the dominant allele. Word 4. Recessive. The recessive allele is the quiet one. It only shows when no dominant allele is present. When both alleles are the recessive version, we always write recessive alleles as lowercase letters. Lowercase b for blue eyes. Lowercase means quieter. Recessive means hidden. Unless it has the space to show. Word 5 and 6. Genotype and phenotype. This pair causes more lost points on biology tests than almost anything else in genetics. Your genotype is your actual allele combination, the letters themselves. BB, BB, or BB. It is what is written inside your DNA. Your phenotype is what actually appears on the outside. Brown eyes, blue eyes, tall or short. Genotype is the hidden code. Phenotype is the visible result. They are not the same thing. Words 7 and 8. Homozygous and heterozygous. If both your alleles for a gene are identical, BB or BB, you are homozygous for that gene. If your two alleles are different versions, BB, you are heterozygous. Heterozygous individuals often look like the dominant phenotype on the outside, but they secretly carry the recessive allele inside. Geneticists call them carriers, and carriers are exactly how two brown-eyed parents can have a blue-eyed child. Let us build the square now, and then come back to prove it. Step 1. Identify your cross. We will use the simplest and most tested example. A monohybrid cross. One trait, two heterozygous parents. Both parents are BB. Both carry one dominant B allele for brown eyes and one recessive B allele for blue eyes. We write the cross as BB times BB. Step 2. Draw a 2x2 grid. Just 4 boxes, arranged in a square. Nothing complicated yet. 2 columns across, 2 rows down, 4 empty boxes inside. This is your entire workspace. Step 3. Write the first parent's alleles along the top of the grid. The first parent is BB, so B goes above the left column. B goes above the right column. Then write the second parent's alleles along the left side of the grid. B goes beside the top row. B goes beside the bottom row. Step 4. Fill in all 4 boxes. Each box receives 1 letter from its column combined with 1 letter from its row. Top left box, B from the top, B from the side. That box contains BB. Top right box, B from the top, B from the side. That box contains BB. Bottom left box, B from the top, B from the side. That box contains BB. Bottom right box, B from the top, B from the side. That box contains BB. 4 boxes, 4 possible offspring combinations, each one equally likely. Step 5. Read your results. Look at what you have in the 4 boxes. 1 BB, 2 BB, 1 BB. That is your genotype ratio, 1 to 2 to 1. 1/4 of offspring will be BB, 2/4 will be BB, and 1/4 will be BB. Now read your phenotype ratio. BB has a dominant allele. It shows brown eyes. BB also has a dominant allele. It also shows brown eyes. Only BB has no dominant allele. It shows blue eyes. 3 boxes show brown, 1 box shows blue. That is your phenotype ratio, 3 to 1. 3 to 1. This is Mendel's famous ratio, the one he found in thousands of pea plants, tall to short, yellow to green, round to wrinkled. Always 3 to 1 when 2 heterozygous parents are crossed for a single trait. A monk in a garden in the 1860s found a mathematical law of nature. Now turn those boxes into probability. Each offspring has a 25% chance of being BB, a 50% chance of being BB, and a 25% chance of being BB. These are probabilities, not guarantees. Each child is an independent event. 3 brown-eyed children in a row does not make the 4th child more likely to have blue eyes. The square tells you odds, not certainties. Now, back to our opening question. 2 brown-eyed parents, a blue-eyed child. How? Both parents must be heterozygous. Both are B-free. They look brown-eyed because the B allele is dominant. But each parent secretly carries a recessive B allele. And when the B from one parent meets the B from the other, the result is B-free. Blue eyes. It was always there, hidden in the code, waiting.
3 mistakes.
These appear every year, and they cost students easy points.
Mistake 1.
Capitalisation. A capital B and a lowercase b are not the same allele. They are different things. BB means homozygous dominant. BB means homozygous recessive. Always write the dominant letter first. Always keep capitalisation consistent. One careless switch and your entire answer is wrong.
Mistake 2. Leaving a box empty.
A 2x2 Punnett square always has exactly 4 boxes. Fill, or, 4. Students who accidentally fill only 3 get the wrong ratio and the wrong answer. Before you move on, count your boxes every time.
Mistake 3.
Mixing up genotype ratio and phenotype ratio. For a B-bat times B-bat cross, the genotype ratio is 1 to 2 to 1. The phenotype ratio is 3 to 1. They are not the same number. Read the question carefully. Know which ratio it is asking for before you start counting.
Genetics does not always follow the simple dominant recessive pattern.
And your exam will almost certainly test you on at least one exception. Let us look at the most common ones briefly. In incomplete dominance, neither allele fully overrides the other. A red-flowered plant crossed with a white-flowered plant produces pink flowers. A blend. Neither the red phenotype nor the white phenotype appears. Something entirely new appears instead. In this case, the phenotype ratio is still 1 to 2 to 1, matching the genotype ratio.
In codominance, both alleles express themselves completely at the same time.
No blending, both fully present. The clearest example is blood type. If you inherit an IA allele from one parent and an IB allele from the other, you do not get a blend. You get both, AB blood type. Both markers appear on your red blood cells simultaneously.
And if your exam shows a Punnett square with X and Y chromosomes,
that is a sex-linked trait. Males carry one X chromosome and one Y. Females carry 2 X chromosomes. A recessive allele carried on the X chromosome will express itself in any male who inherits it, because he has no second X to mask it. This is why conditions like color blindness appear far more frequently in males than in females.
For the dihybrid cross, where you track 2 traits at once, you draw a 4x4 grid with 16 boxes.
2 double heterozygous parents give a phenotype ratio of 9 to 3 to 3 to 1. 9 shows both dominant traits, 3 shows only the first, 3 shows only the second, and 1 shows both recessive. That 9-3-3-1 ratio is the signature of a dihybrid cross. Recognize it immediately.
One rule for your exam above everything else.
Draw the square. Do not try to solve genetics problems in your head. Every time you write out the grid, label the alleles along the top and side and fill in all 4 boxes carefully. You protect yourself from errors. The square does the thinking. You just set it up correctly.
Let us bring it all together.
A Punnett square predicts the possible genotypes and phenotypes of offspring from a genetic cross. It applies Mendel's law of segregation. Each parent contributes 1 allele to each offspring. A monohybrid cross of 2 heterozygous parents gives a 3 to 1 phenotype ratio and a 1 to 2 to 1 genotype ratio. A dihybrid cross gives a 9-3-3-1 phenotype ratio. Exceptions include incomplete dominance, codominance, and sex-linked traits.
For your test, 3 things to lock in.
1. Set up the grid correctly. Parent alleles along the top and side. Fill all 4 boxes. 2. Know the difference between genotype ratio and phenotype ratio. 1:1 versus 3:1 for a monohybrid cross. 3. Know the 3 main exceptions: incomplete dominance, codominance, and sex linkage. Those 3 points cover most genetics questions you will face.
One way to keep dominant and recessive straight. The dominant allele is the loud one. It speaks over everything else. Write it with a capital letter. The recessive allele is quiet. It can only be heard when there is nothing louder beside it. Write it lowercase. Loud letter, quiet letter. Every time you see the word dominant, think uppercase. Every time you see recessive, think lowercase. 2 parents, both with brown eyes. A child with blue eyes. What seemed impossible becomes obvious the moment you build the square. 2 quiet B alleles hiding inside both parents finally meeting. The answer was always there, written in the code. You just learned how to read it. If this finally made Punnett squares click for you, leave a like so more students can find it. Subscribe because your next genetics topic is on the way, and we will make that one just as clear.